Hello, everyone, and welcome to our switchSENSE webinar, Antiviral Discovery Using switchSENSE, Advances in Polymerase Characterization for the COVID-19 Era. My name is Isaac Plant. I'm the Technical Sales Manager for Dynamic Biosensors in the Western United States. Joining me is Gansha Bulbul, who is our Field Application Specialist for the territory. And we're excited to talk to you today about switchSENSE and polymerases. So to give you an outline of what we'll be talking about, we're going to be going through viral polymerases and the role they play in RNA virus infection. We'll be giving an overview of current biochemical methods for studying polymerase activity and how they are preventing and limiting RNA-dependent RNA polymerase inhibitor discovery. We'll go through then the switchSENSE technology and how it offers a comprehensive platform for assaying polymerase activity, studying the binding kinetics and mechanism of action of RNA-dependent RNA polymerase inhibitors, and then move into how these simplified assays using switchSENSE can help you advance drug development and bring these inhibitors to the clinic. So just briefly, switchSENSE is Dynamic Biosensors' proprietary technology for studying biomolecular interactions. It's based on a DNA nanolever that's electro-switchable and allows us to study everything from kinetics, affinity, avidity for interactions, and then conformational changes of proteins and thermodynamics of these systems. Today we'll be focusing on proteins and DNA RNA interactions, and particularly polymerization of DNA RNA. And the reason we're going to focus on that is because this polymerization by RNA-dependent RNA polymerases is key for a number of human viruses. And so these viruses that depend on RdRPs, from rhinoviruses, so relatively low risk, right, colds, to much more dangerous diseases such as Ebola. But all these viruses you see proceed through RdRP's steps, and without treatment, obviously, some of them can make you quite sick, which is why inhibitors for RdRPs have been developed, targeting drugs, particularly hepatitis, Ebola. So, and one of the ones that's been much more in the news recently is the SARS-CoV-2 virus, the causal agent for COVID-19. And this, like other viruses, proceeds through an RdRP step. And this is why the first drug to show efficacy against COVID-19 is remdesivir from Gilead, which is an RdRP inhibitor. An RdRP inhibitor that's actually been studied for hepatitis and Ebola previously, but showed lower efficacy. But now this is the first drug for COVID-19 that's showing some efficacy. But again, we'd obviously like to see more of these come to clinic, but there is a limit on the biochemical side. And those are because the current biochemical methods have some problems. So the classical way to study polymerases and activity is using radiolabeled nucleotides, obviously very high sensitivity, but because they're radiolabeled, they're difficult to work with. They have limited throughput, and without significant assay development, you have a problem studying the mechanism of action of any inhibitors of polymerases. The advance, or the next step often from radionucleotides, is using fluorescently labeled nucleotides or intercalating dyes. These are much higher throughput for studying polymerases, but they have much lower sensitivity than radiolabeled. They can also interfere with the polymerase, changing the rates of elongation, and which can give you artifacts when you try and study inhibitors. These also, like radiolabeled nucleotides, don't provide mechanism of action of inhibitors easily or without further assay development. You can do some more kind of complex experiments by using fret-labeled polymerases, by putting fret pairs on polymerases to get mechanism of action inhibitors, but this requires labeling the polymerase, which often isn't an option, and requires single molecule techniques. So to fill this gap, we've taken our switchSENSE technology and really developed polymerase applications that allow for higher throughput, allow you in single experiments to understand the mechanism of action of inhibitors, and does so in a label-free, highly sensitive way. So with our helix biosensor and switchSENSE, you would be able to potentially in single reactions measure the K-on of the polymerase, so the initiation, the elongation or K-catalytic rate, as well as the dissociation of the polymerase. And by being able to observe all of these rates in single experiments, you can really do high throughput screens of inhibitors while understanding their mechanism of action, which you couldn't do with previous biochemical techniques. And with that, I will hand it off to Gansha, who will be able to give you some more of the science about this.
Thank you, Isaac, for the introduction. And in the following slides, I'm going to explain how we can use our switchSENSE technology for polymerase characterization at each step. I would like to start with introducing the experimental workflow for real-time elongation measurements using switchSENSE. We start with our standard biochip, which is functionalized with a single-stranded DNA that carries the reporter fluorophore at the distal end for real-time observation of polymerase binding and primer elongation. Then we functionalize the surface using a small amount of primer, which enables us to work with low concentration and quantities of DNA with a low density surface. We can vary the length of the starting primer, which effectively impacts the elongation time. For example, we can use a short primer for fast polymerases so that we have a readout involving more data points before the primer is fully extended. After we form our surface and fully saturated, then we can associate the polymerase. Here we can study the kinetics of binding and figure out the K-on rate. Afterwards, the kinetics of elongation rate can be studied by introducing the NTPs to the solution. And with switchSENSE here, we can study the K-on and elongation rate separately. This enables us to study various different aspects of mechanism of polymerase activity, and we can figure out how an inhibitor can potentially interrupt that. Because we can also dehybridize the double-stranded DNA, the individual chip is not dedicated to only one experiment. So you can regenerate the surface and use this multiple times over and over again for different types of experiments. With switchSENSE, we have an automated experimental workflow for surface functionalization, binding kinetics, and elongation activity. We employ our molecular ruler principle for investigation of DNA elongation kinetics by polymerases. Here, we are using a fluorescence-based technology. Therefore, our readout is fluorescence. The intensity of the fluorophore that we have on our surface is dependent on the distance to the cold surface. The template for the DNA elongation is a double-stranded DNA nanolayer that is modified with a single-stranded overhang. In the initial stage, the partly single-stranded nanolayers exhibit a relatively low fluorescence intensity owing to their high flexibility that allows them to approach the quenching of the cold surface. When the polymerase recognizes this double to single-stranded DNA break in the DNA, it binds to the strand, and then we can monitor the K-on rates. Then you can flow in the NTPs as a secondary step which allows polymerases to begin elongating the DNA. And we can measure this elongation as an increase in the fluorescence as the double-stranded DNA becomes more rigid and moves away from the surface. And when the elongation is finished, the fluorescence is stabilized. Now that we look through the experimental workflow and the principle of the measurement, I would like to show you some exemplary data in the following slides. In our first example, I'm going to show you the work we did with T7 RNA polymerases and how we monitor the DNA binding and transcription activity. The first step of studying the polymerase activity is to associate it. After the association of RNA polymerase to the overhang, a decrease in the fluorescence intensity can be observed. The data was further analyzed using a global mono-exponential fit model, and we were able to quantify the K-on rate. And after the addition of NTPs, then we can observe the elongation as an increase in the fluorescence signal and calculate the activity rate. In addition to monitoring the polymerase binding and the elongation, we can also do further characterization. Different assay parameters which are important to study for polymerase activity can be addressed by using switchSENSE. These include varying the temperature and varying the dNTP concentration, as illustrated in this slide for DNA polymerase. Since our technology and the readout is very sensitive, we can easily change the concentration of the NTPs. The figure on the left shows the elongation monitored in real time by the fluorescence that is emitted from the standing DNA for different dNTP concentrations at room temperature. Furthermore, the elongation activity was analyzed in real time as a function of temperature from 5 degrees Celsius to 45 degrees Celsius, as shown in the figure on the right. So if you are working with polymerases that are temperature sensitive, we can modify the temperature on the switchSENSE surface, and we can monitor the changes on the activity rate by following the fluorescence changes from 0 to higher values. And then by changing the experimental parameters, we can actually modulate the activity of polymerase. This ability is valuable when you are working with very fast polymerases, typically for the purpose of slowing down the readout and increasing the measured data points before it completes the full template elongation. After we have reviewed the standard assay setup, as well as explored the parameters that can be varied for the determination of binding kinetics and elongation activity, we will move forward to describe how this can be applied within the workflow of antiviral drug discovery. Usually the objective of antiviral discovery is to be able to see if the presence of small molecule inhibitors interfere with the K-on rate or with the incorporation rate of NTPs. As opposed to conventional in-solution assays, where you would need a readout requiring labeling, in our case, the readout is the fluorophore, which is already on the template DNA. Therefore, it is easier to make a correlation with the polymerase activity with the readout. With the advantage of switchSENSE technology, you are given the possibility to work with unmodified nucleotides. But if you like, you can also work with chemically modified nucleotides and monitor how that changes elongation rate in real time. You can see the successful application of switchSENSE from the figure on the right to monitor incorporation of NTPs and how the presence of chemically modified nucleotides affect the rate of fluorescence change. You can see in this slide from the bar graphs that the natural nucleotides are normalized to 100%. Then there is a significant reduction in the rate of incorporation when working with other chemically modified nucleotides. We observed this behavior for both cyclopropynylated and triazine-modified nucleotides. In both of the cases, both artificial nucleotides, adenine and uracil, are incorporated and the elongation rate is reduced with a modified uracil having a stronger effect. In this slide, we would like to show you how does the readout change when we are actually adding the inhibitor. We can determine the impact of the inhibitor if it is made on the binding affinity and or catalytic activity. Here from the figure in the middle, you can see the dose-dependent impact of adding an inhibitor to our system. It leads to a reduction in the elongation activity of the polymerase, which is followed by monitoring a decrease in the fluorescence with concentration-dependent manner. Therefore, we can also determine empirically the IC50 of inhibitors. And with that, I would like to hand the presentation over back to Isaac to give the concluding remarks.
Thank you, Gansha. So now, to give you an overview of what we talked about, we went through viral polymerases and how RdRP polymerases play a critical role in infection for a number of diseases. How the current biochemical methods for studying polymerase activity are lacking, both often in their throughput as well as the depth of readout, preventing an understanding of the mechanism of action of a number of inhibitors. Gansha walked us through how switchSENSE is really a comprehensive platform for studying polymerases inhibitors, how it can be used to measure all the characteristics of polymerase activity, it can be used for screening inhibitors, and for determining the mechanism of action of inhibitors, all within a single experiment. And this really allows for simplified assays and drug development that can reduce clinical or attrition of drug candidates in clinical stages, because you have artifact-free data about the inhibitors and really high-quality knowledge about their mechanism of action. So finally, I'd like to mention that the switchSENSE technology the current implementation is within our helix line of instruments. So we have instruments that are suitable for any size organization or need, ranging from the Helix Plus, which incorporates all of this in a single instrument, up to our double helix bundle and 4 helix bundle, which allow increased throughput and modularity of allowing you to run multiple experiments at once. Finally, we'd just like to thank you for joining us, and we'll move into a discussion and a question and answer session with Joanna. Thank you very much.